Swayam Prabha. Digital India. Educated India.
Dear students, welcome to the organ printing course. Let's start our first lecture, that is Introduction to Bioprinting. So before going into actually telling you what is bioprinting, let us start with where exactly this bioprinting fits in the whole scheme of tissue engineering and regenerative medicine. So if you see, the basic principle of tissue engineering is to generate artificial tissues and organs for repair, regenerate, augment the diseased or damaged tissue. So in this process, the first step is, because our interest is to you develop artificial tissues for a particular patient, then we start with isolation of cells from that particular patient. So there we can use different techniques, but biopsy is one such technique where we collect a very small amount of tissue from the patient's that particular tissue, and then we use that biopsy tissue for cell isolation. So we isolate the cells, isolate the target cell basically from that biopsy sample, and then we use them for expanding. So the cell number is expanded using standard cell culture techniques, using standard tissue culture plates, providing all necessary conditions like nutrients, even nutrient and oxygen. All these things are provided, so the cell number increases. So that is the expansion of cells. And when the cell number reaches to a certain level, so that we can use them for further use, then you seat them on a substrate, or it's also called a scaffold. Different 3D scaffolds are being used for cell seeding, and then we, after culturing them on the cell seeded on the scaffolds for certain time period, then that the cells further grow in numbers, they migrate within the structure, within the scaffold, and then they produce or lay down extracellular matrix within the structure, so that the graft is being produced. So that is the generation of graft. Then that graft, after maturation, we can actually go for implantation of this graft on the damaged or diseased tissue, so that then it will help or facilitate the tissue regeneration process. So this is the whole scheme of tissue engineering, and here the bioprinting fits in, where we actually trying to develop artificial tissues or organs. Here we don't print separately scaffold and then seed cells, but what we do, we mix the cells along with some materials, the hydrogel, and then we use that mixture, cell plus hydrogel mixture, that is also called bioink, and then we use that for printing artificial tissues and organs. So at this stage we use them for generating this graft. With the generation of graft can be done with the help of this bioprinting process. There are various types of scaffold fabrication methods are available, starting from solvent casting, particulate leaching, phase separation or phase drying, gas forming, wet spinning, electrospinning. Also there are supercritical fluid CO2 products based scaffold manufacturing techniques, and also different additive manufacturing techniques. So among this, in this course we'll be discussing mostly how additive manufacturing techniques can be used for preparation of artificial tissues or organs. So that is called bioprinting. A little introduction about the additive manufacturing or 3D printing. If you know, additive manufacturing or 3D printing is a process of fabricating three-dimensional solid objects using a digital file. That means we design a 3D structure using some computer design software like SolidWorks, Solid Edge. There are many of such things. We design that 3D structure in the using that software, and then we use that digital file created, or the 3D image file created using the software, to print the 3D structure using a 3D printer. So 3D printing is a process by which we develop the three-dimensional object using a 3D printer from a digital file. And then this process, because why it is called additive manufacturing, because this process is an additive process. We add small amount of material and then continuously to make the whole 3D structure. So this is an additive process. And then how the whole structure is printed, basically by adding, as I told already, by adding small amount of material, by we add the materials layer by layer. So first the first layer is printed, then on top of that the second layer is printed. So the successive layers of materials are printed to develop or to fabricate the whole structure. So it's also a layer by layer manufacturing process. Now each of these successive layers can also be considered or can also be viewed as a thin horizontal slice of the whole object. Right? So this is 3D printing. So here mainly is the three steps involved in this process. The first step that starts with the modeling, where we print or where we design the 3D three-dimensional structures of the particular object. That suppose you want to print a particular structure, then you always will start with the designing of the 3D structures, and that designing can be done, as I told, already using some CAD software, that is computer aided design software, and there you can actually create the whole 3D geometry. So that is the process called modeling, where basically we create the 3D models, and also that is the virtual blueprint for printing the whole 3D structure. And then there are various other operations like in 3D printing, because the printer prints in a layer by layer manner. So we, after creating this 3D object, generally we go for slicing that object, so that we create the different horizontal sections or slices. So each slices will be taken at a time by the 3D printer, and that will be printed as a layer, right? So that slicing operation is also a major thing. And then meshing operation, also mesh generation, there is another process by which we can actually create the mesh of the 3D print structure, so that the printer can print them in that fashion. Then the actual printing process is done, where the 3D printers read the design, like whatever you have created, that 3D design, that 3D using CAD software, that design the printer will read that design and then lay down the materials in a layer by layer fashion successively, so that, and those layers will get joined with each other to get so that we can generate the whole 3D structure. The next process is finishing, because most of the time the object that is produced with a 3D printers is not ready for use or for ready for market. So the finishing operation is done there, where the different types of finishing, like removal of the support structure, polishing, finishing, sanding, buffing, coloring, everything, all these things process are done, so that the appearance of the 3D printed structure improves, the user experience will further also improve. Now the next question is whether 3D printing is a bottom-up approach or top-down approach. If you see 3D printing is a additive process, like where we add materials layer by layer to build the whole structure. So we start from scratch to build the whole structure. So in bottom up, actually that is what bottom up process, where we use that to build the whole structure. Top down is when we start with suppose we start with a bigger thing, then we cut down that thing, that the whole block into a 3D structure. So that is top down process. So 3D printing is or additive manufacturing is a bottom up approach. Now let's discuss what are the different types of 3D printing modalities available. And if you see, 3D printing is not a single technology, it's an umbrella of different technologies. And various types of 3D printing technologies evolved over time because all this 3D printing technique, or each of this technique, is capable of printing one type of material, one or few different types of materials. Like if we take example, like fused deposition modeling is a 3D printing technique, where it uses thermoplastic material for printing 3D structure, and it's an extrusion based process. That means it extrudes the material, the thermoplastic molten thermoplastic material, in the form of a 3D structure. Then there are electron beam freeform fabrication technique that uses metal wares as a input material and print the whole structure. Similarly, different types of 3D printers, they are capable of using different other types of materials and then printing different structures. So based upon your application, first you have to see the what type of materials you are going to use and what type of structures you are going to print. Depending upon that, you can select one or other type of 3D printers. So these are different types of 3D printers, 3D printing techniques that has been evolved over time. Those are here listed here. Now, commonly 4 different types of 3D printers are used mostly, and when 3D printers are mostly chosen for an application, that is depending upon the materials to be used and what type of structures you are going to produce. So 4 different types of 3D printing techniques or common 3D printing techniques used for medical applications are stereolithography, selective laser sintering, fused diffusion modeling, and thermal inkjet printing. In this course, we will be discussing what are the printers mostly used for bioprinting applications, and will see that in a later slide. Now let's understand what is bioprinting. Right? We have seen 3D printing. Now what is bioprinting? Bioprinting is also an extension of 3D printing techniques, but here a few things are few differences with 3D printing, like because in our interest is to print artificial functional human tissues and organs. So always we start with the materials that is the building block of any tissues or organs, like the cells are the building block of a tissue, so we start with cells, right? So we use living cells along with some biologically relevant materials or bioactive factors to print the human tissues and organs, and this is also layer by layer process. So we layer by layer fashion, we deposit these living cells along with biologically relevant materials with or without bioactive factors to develop the artificial or functional human tissues and organs. So this process is called bioprinting. It's an semi-automatic or semi-automatic process. The process of this bioprinting is also can be divided into 3 steps, like pre-processing, processing, and post-processing. In pre-processing, we do all this is required for printing a tissue structures, like because now suppose we are targeting a particular tissue structure, we want to print that, then always we don't start here with CAD modeling of the tissue. What we do, you use different bioimaging modalities, because all of these tissues are unique. There's my structure, microstructure, size, shape, everything is unique. So we start with different bioimaging modalities. So we capture the image of that particular target tissue, and then we can get the 3D image of this particular tissue structure using with this bioimaging model like CT and MRI. We get the 3D image, and those images are mostly available in DICOM formats. Then we convert them into STL file. So STL file, because that STL file is mostly understood by the most of the 3D printers. So that is the 3D geometry we design, or that is the CAD file we generate from this imaging, from the 3D images, and that is the pre-processing steps. After that, the actual printing process is done. So there is processing where we develop the bioink first, and bioink, as I already told you, bioink is an printable formulation consisting of cells, matrix materials, bioactive factors. So we develop the bioinks using a combination of all these different things, and then it is those after the bioink is developed, that is loaded to the bioprinter, and also here from here the CAD model is also loaded to the bioprinter. So the bioprinter will now use this bioink to print the 3D structure using the design that is created, or the 3D image or the STL file that is being loaded. So then we'll be able to print the structure, and but when we print a 3D structure using a bioink, soon after printing, it is not called tissue. It is called pre-tissue because at this stage it is just a cluster of cells plus materials and bioactive factors. So the function is not yet established within this tissue structure. So that's why we don't call them tissue, we call them pre-tissue. Now upon maturation, when we provide all necessary conditions like nutrients, oxygen, all this thing, then this the cells here, they will proliferate, migrate, differentiate, and also they will grow. They then the also the whole function of this tissue or the specific functions that is required for the particular tissue will develop. At this stage that can be they are matured, and then we can go for implantation to a particular site. So that is the diseased or damaged site, right? So that is post processing. Now the main steps, as I said, the main steps of 3D bioprinting is first pre-processing, where we generate the CAD file using bioimaging modalities like CT, MRI, all these, and then that is those are the blueprints for printing 3D structures. So that is the, and here that is basically here we create the pre-processing. Also in the here and then the actual processing or the printing, where we create we generate the bioink, we do the actual printing using that CAD file, and then solidification of the structure, printed structure or match, that is also important for stabilization of the for the stability. After that we do post processing. In the post processing step, that is where we actually mature the tissue maturation happens. There we can culture them in some bioreactor, providing all necessary conditions for their survival, and also we can further also use some conditions for accelerated tissue maturation. So the steps involved in this organ printing are as follows. Like, first you have to select the target tissue or organ, right? That is the very first step. Suppose your interest is to print a particular target tissue or organ, so that we can actually basically repair or regenerate the damaged or diseased tissue. So we first we select that particular tissue. That is the target tissue or organ. That is, you have, suppose that you have selected them, then you can take an image. That means you using the bioimaging modalities like CT and MRI, we take images or capture images of that particular tissue in 3 dimensions. That is means the different slices, right? So then from that we generate a blueprint using different CAD software, so that we can get the 3D image data or the virtual data of this particular tissue or organ. Then you prepare the bioink. As I said, bioink is a printable formulation consisting of cells plus materials and also bioactive factors. So we prepare the bioink, and this bioink is very specific for a particular tissue. Like, suppose your interest is to print a skin, then you will be developing a bioink that is suitable for printing that skin, like that is a skin specific bioink. Then so the bioink is prepared, and then after the bioink prepared, the next step is the actual bioprinting, where we use this bioink using the virtual blueprint, that is the design. The bioprinter will print the tissue structure, and then we allow them to solidify or stabilize the tissue structure, and then after that it goes for maturation, where the tissues will be, or the printed structure will be mature, so the cells will grow, they will produce extracellular matrix, lay down the extracellular matrix, and in this process also the biomaterials that we use, mostly those are biodegradable materials, so they will start degrading, and so the tissue will get matured, and then it can be used for different applications, including implantation to the target tissue or organ structure. Let's see what is the state of art in this technology. That means what are the current results and achievements over the years, right? So the very first attempt of bioprinting or 3D printing was done by C.W. Hall in 1984. Then he used a technique called SLA, that is stereolithography technique. He invented that technique, and that was it in 1984. Then in 1996, Dr. Gabor Forgax and his colleagues, they made an observation that the tissue or the cells have liquid-like properties. That means, suppose if we print a cells, if cluster of cells, then the cells can able to migrate and they able to organize themselves into a particular tissue hierarchy. So the cells, and they have seen this during embryonic development, also during wound healing. This tissue like liquid-like property cells have this liquid-like property. They stick to each other and then they organize. In 2003, a very important breakthrough happened when Dr. Thomas Boland, his lab basically modified a HP inkjet printer to print cells. So that was the very first attempt of printing cells with a modified bioprinter. And in this case, a modified HP inkjet printer. And also, the same year, another important breakthrough happens when scientist named Dr. Anthony Atala from Wake Forest Institute of Regenerative Medicine, they developed a synthetic scaffold seeded with the patient's own cell to replace the urinary bladder of that particular patient. Now, this is a very important breakthrough because the patient survives after this with this synthetic scaffold that is seated with his own cells. So that is the urinary bladder, artificial urinary bladder, and then that he was doing fine after that. Now in 2004, Invision Tech, they have created their first extrusion-based bioprinter named 3D Bioplotter. And then also in 2004, Dr. Gabor Forgas, he developed a new technology based upon his observation that tissue has this fluid-like properties. So he engineered 3D tissues with only cells. Then Organovo is an American company. They have created their first bioprinting technologies, bioprinters using Forgas technology. Their name is Novogen MMX. Then in 2000, after that, there are series of discoveries or at a series attempts where bioprinting technology was used to develop one or other tissues. Like in 2000, there are some examples I am giving here. So in 2011, fast cardiac sheet or patch was developed. Then after that, fast lung tissue patch was developed, and then Organovo again created a in vitro liver model that is used for drug and toxicity screening. And then they have supplied this to different pharma industry for testing their drugs to check whether the drug has any toxic effect on the liver or not. That liver is an in vitro liver model. Then in 2019 or 2019, another important breakthrough happens. A few researchers from Tel Aviv University, they have bioprinted a four-chamber heart or a human heart. They have printed and they have shown the possibility of developing an artificial heart with the help of a bioprinter. After that, there has been various attempts to print different other tissue structures, and also in the same time there has been a few attempts where researchers from various across the globe have used their bioprinter or they have done the animal trials of their bioprinted tissues, organs, structures. So the evolution of tissue and bioengineering as follows. So before 2015, like in the past 2015, mostly the researchers were printed small tissue size, and for that, and those are mostly used for different in vitro drug screening or toxicity screening, and few other small tissue structures. So mostly various proof of concepts were developed. And then in recent past, like in starting from 2015 to 2020, simple tissues were being bioprinted, like those are like cardiac patches, segments and tubes, small blood vessels, all these things were bioprinted. And then at present, like in this decade 2020 to 2030, researchers are mostly trying to develop different tissue structures, but like liver lobules, kidney structure, skin, little large skin patches, and then the different other tissue structures or tissue or organ structures. But if you are thinking of a full organ like kidney or complex organs like kidney, liver, then those are not yet fully developed, or those are not bioprinted till now, because those organs are very complex. So researchers hope that maybe by 2030 or after 2030 we'll be able to develop, or we'll be able to bioprint the whole complex tissue structures, or that will be functional, so that can be used for implantation, because there are many patients waiting for artificial tissues or organs, because the demand, there is a the the demand is far more than the actual supply. So that's why we need to develop artificial tissues and organs to save patients. And by we hope that bioprinting is a promising technology, and definitely it has the prospect to prospect to fabricate artificial tissues and organs. So, but at this moment, the complex organs are not being bioprinted. Now let's discuss about the different bioprinting approaches, or what are the different types of bioprinting modalities available. As I discussed in 3D printing, like there are 4 different common types of 3D printers available. Similarly, if we again classify the bioprinting technologies, there are mostly 3 types: inkjet-based bioprinting, extrusion-based bioprinting, and laser-assisted bioprinting. Now, each of these bioprinting techniques, they have their own advantages and disadvantages. But what types of bioprinting, or what type of bioprinting technology is suitable for a particular application, that is always depending upon the application, depending upon the target tissue structures. And what that means, what type of tissue or organs you are interested to bioprint, or you are targeting to bioprint, and what is the material that is going to be used for bioprinting that particular tissue or organs, and what are the different types of cells that will be used for bioprinting of the particular tissue. So all this, so using of a particular 3D bioprinting technique depends upon all this.